Okay, so this is your Unit 2 review, and I'm having to record it through Zoom, so I hope this works. All right, so energy and enzymes and cellular respiration and photosynthesis are the topics for Unit 2. And so just to remind you, when we talk about energy in this class, we're talking about the delta G, which is the Gibbs free energy. And it's really just referring to the delta is a change. So it's referring to the change of energy due to a chemical reaction. And remember that when you calculate delta G is always the energy in the products minus the energy in the reactants. So this little table is just to remind you about our two types of reactions. So we have exergonic, which is energy releasing reactions, and endergonic, which are reactions that require energy. So energy releasing reactions would also be considered catabolic, right? So when you're breaking down food, you're releasing energy for your cells to use. Anabolic requires energy. So this is when you're building things up, right? And so you can tell it's an anabolic or endergonic reaction because it has a positive delta G. That would tell you that the products have more energy stored in them than the reactants started with. So that means you have to put energy into this reaction to make it happen, and vice versa. On the exergonic, it's a negative delta G because it's releasing energy. Both of these can use enzymes to reduce the activation energy, and we'll talk about that in just a second. Remember, here's our pictures of enzymes. The first one on the left is showing a catabolic reaction. So it's taking the molecule and breaking it down and releasing energy. Over here we have an anabolic, where we're taking two substrates or two products and putting them together. And so we would need to put in energy. And these gray blobs are our enzymes, right? So remember that enzymes are proteins. And the way they reduce activation energy and the way they act as a catalyst, which means they speed up the way they do that is they are bringing these substrates together and they're bending and twisting them to allow bonds to be broken and remade. Also remember that enzymes, are reused over and over. So they can do their job and then grab new substrates and do their job and grab new substrates. The other thing to remember is that enzymes are very specific. Right? So each enzyme has one job, and you saw that when we looked at the stages of glycolysis. I think there was 10 different enzymes that it took just to take one molecule of glucose and basically break it in half. Excuse me. A couple other words you learned with respect to energy and enzymes and reactions. Anabolic goes along with dehydration synthesis. Right? So you're using energy, you're taking smaller molecules and you're making a larger macromolecule. So this could be, for example, hooking amino acids together, right, to finally make a protein. And so hydrolysis is just the opposite, where you're taking that protein and breaking it down to amino acids. Remember that hydrolysis means that you use water to lyse or break the bonds. And dehydration synthesis means that as you're making these bonds, you release water. So water is made, water is used. Okay. So having a chart reminding you of those different properties would be helpful. So let's take a look at some images and see if you can apply your knowledge. So the first question, excuse me, is what type of reaction is shown? Okay. So a couple ways to tackle this. You can see that the products, oops, have more energy than the reactants. They're higher up there. So if you have a big number minus a little number, just say 10 minus 1, that's going to give you a positive number. So the delta G is positive, and we know that means it's an endergonic reaction, right? So you can see it's taking energy. You need to put some energy in here to make those products. Okay. So I guess I kind of, answered this. So we figured out that the delta G was positive. I want you to calculate it based on this, excuse me, graph. And so what you're going to do is you look across and you say, okay, the products have a value of 15 and the reactants have a value of 5. And so we get delta G equals a positive 10. So remember, if you're analyzing one of these graphs and you forget, you can always make up numbers and calculate your delta G, and then you'll see when it's positive, it's an endergonic reaction, and when it's negative, it's a exergonic reaction. Okay, the next question asks, which line shows the effect of an enzyme? And hopefully you realize that it's the red dash because enzymes lower the activation energy. So what is that activation energy? And the activation energy is always from the reactants to this peak. And remember that the activation energy is that little extra oomph that is required to strain and make those bonds break and remake. So it doesn't have anything to do with the molecule's energy in the molecule itself. It's that little extra oomph that's required. And you can see that it's much lower when you have an enzyme. So that's how enzymes reduce the activation energy. All right, so I want you to try this on your own. Draw an exergonic reaction, label the delta G, label the EA. So go ahead and pause. And then when you come back, hopefully you drew something like this. Right, so it's energy, it's the reaction taking place along this x-axis, and you're going to start off with more energy in the reactants, less energy in the products, and then when you draw that graph, you just give it a little hump and then connect the lines. The delta G is always the difference between the products and the reactants, so that's the delta G, and the EA is always the reactants to the hump. So if you were going to draw the effect of an enzyme, you would just draw this everything the same and decrease that hump. Okay. So that's an exergonic energy releasing, right? It has a negative delta G because the product, say, is a value of 2, and the reactant has to be bigger, value of, say, 10, and that would give you a negative number. So which type of reaction would require ATP, an exergonic or an endergonic reaction? And the answer is an endergonic reaction, right? Because this one is releasing energy, but the one up here required energy. So that's going to require to be coupled with ATP or the breakdown of ATP. So let's look at that. So remember, ATP is our energy molecule in the cell, and we have hydrolysis, so we're using water to break a bond, and we're breaking this last phosphate bond. And energy is released, about 32 kilojoules per mole. So you don't need to know this number, I just want you to see that it has a negative delta G. And so this energy that's released is used to make endergonic reactions happen. So energy coupling is used to make endergonic, energy requiring reactions happen. Okay, so let's look at energy coupling. So here's a reaction. We want to make sucrose. Sucrose is table sugar, and that's done by combining glucose and fructose. But that's not going to happen on its own because it requires an input of energy. So if we couple it, if we put it together with ATP, the breakdown or hydrolysis of ATP provides the energy needed. And you can see that ATP is going to release 32. You only needed 26. We get some heat made. That's how we what happens to extra energy from a reaction is it's given off in the cell as heat. We can't use heat for work, so the cell tries to be efficient and couple reactions together. But energy coupling, again, is how you take a reaction that needs energy and endergonic, and you combine it with a reaction that releases energy, which is exergonic, and the enzyme can then make sucrose, make sugar. Remember that ATP is used and made, right? So again, this is showing you when you break down ATP, you release energy. And remember, the breakdown products of ATP is ADP, which means it has 2 phosphates. ATP means 3 phosphates. And then you've got to use energy to remake ATP, right? And that's phosphorylation. This adding a phosphate group to ADP is called phosphorylation. And we know that we can make lots of ATP 2 ways: oxidative phosphorylation and photophosphorylation. No, that's not very good. Photophosphorylation. I don't think I quite broke up that word correctly. And remember, we're going to make ATP using the energy from the movement of electrons. We're going to talk about electron transport chain. We're going to talk about a hydrogen ion gradient and chemiosmosis. Excuse me. So let's switch gears now and first talk about cellular respiration. So I highly suggest you pause the video here, write the equation for cellular respiration, draw mitochondria, Label the parts, label as much as you can. Sometimes we call this brain dump. This is a really great way to see what you remember, and that way you know what you still need to study. I'm going to continue on with the video, but again, best way to study is to get things out of your brain. Excuse me. So here's the chemical equation for cellular respiration. You need sugar, you need oxygen. This is aerobic respiration. And you're going to produce some byproducts, carbon dioxide and water. But the most important is you produce a lot of ATP that the cell can use for other chemical reactions. Hopefully you drew your mitochondria. You remembered that it had an inner and outer membrane. You might have labeled the folds of the inner membrane as cristae. Right? The matrix is that space inside the cristae or inside the inner membrane. And then the intermembrane space, the space between the two membranes, is going to be important for generation of our hydrogen ion gradient. So if you were labeling where cellular respiration, the steps happen, remember that glycolysis happens in the cytosol, so outside of the mitochondria. So here's where our first reactant, glucose. And we're not going to go through the details for your final exam. What I want you to remember is big picture, where things happen, where the products and reactants are used and made. We have the Krebs cycle, right, happening. And this should really be happening inside the matrix, right? Just realized that's not a great picture. And we have the electron transport chain happening on the inner membrane. This is the outer membrane. And so we talk about making a little bit of energy, but the most of the energy is made from that electron transport chain. So let's look a little bit deeper detail. So again, excuse me, here's the matrix showing the Krebs cycle, reminding you that as we break down, glucose, we're releasing CO2. Here is where oxygen is required as the final electron acceptor. Here are our electron carriers, super important. Remember that they are made in glycolysis and conversion of pyruvate to acetyl-CoA and the Krebs cycle. And these electron carriers are what's so important to bringing the electrons, the excited electrons. And when I say excited electrons, I mean electrons with energy. And it brings it to this whole thing. This is your electron transport chain, your ETC. And so as those excited electrons move, right, they release energy and they make this hydrogen ion gradient. So you see here, high H+ concentration. We can call this a proton motive force. We can call this an electrochemical gradient. But what's most important is that we're using electrons in the electron transport chain to make a hydrogen ion concentration gradient, which then is going to make ATP. So this blue molecule is ATP synthase, right? And remember, as the hydrogen ions flow down, ADP and phosphate are cooked and we make ATP. So this big process is called oxidative phosphorylation. Oh, and the last thing, don't forget, this is where water is made. So you should be able to look at your overall chemical equation and know where each of these reactants are used and each of these products are made. Okay, I think that's all I want to remind you about cellular respiration. Let's do a few clicker questions. So I would suggest you just pause the video, and there are, I believe, 3 clicker questions. Go through and answer them on your own and then come back to the video. So hopefully, again, you're looking at a PowerPoint on your own, or just pause between each question. So this first question says, a person holds their breath without oxygen. What part of cellular respiration would be affected first? And the answer is the electron transport chain. Because remember here, we need oxygen to allow those electrons to flow out of the electron transport chain so we can keep putting more and more in. So if you stop the electron transport chain, then you can't make hydrogen ion gradient, and then you can't make ATP. Without sugars, such as glucose, cellular respiration will not happen. True or false? And that answer is false, because I want you to remember you don't just eat sugar, or hopefully you don't, right? You eat proteins and you eat lipids. So we have lots of different sources of energy from our food. We just talk about the cellular respiration pathway with glucose because that's the best studied. But if you get into a biochemistry class, you might learn where proteins and lipids, things like that, come in to also be broken down to provide or to help make ATP. And this is a challenging one. I want you to really think about it. And my suggestion is you go through all the answers and say why or why not they would be correct. So you isolate mitochondria from a cell, put them in a beaker of water. What could you add to the beaker to stimulate the mitochondria to make ATP? So my answer that I picked was NADH, right? The mitochondria cannot use glucose. Glucose is broken down in the cytoplasm. Oxygen is found in water. So that's why I say that one's not correct. These two are byproducts. They're not needed to make ATP. They're released in the production of ATP throughout this whole thing. So NADH, remember, is the electron carrier to keep that electron transport chain going. If you had to pick, if this was a free response, what could be a better answer for this question? I think maybe a better answer would be you would provide pyruvate, or you would provide acetyl-CoA, right? Something that you lost in glycolysis, because remember, In the cell, glycolysis is happening out here, and the product, pyruvate, is going in here to feed the Krebs cycle and mitochondria. So that's what I'm just trying to get you to think about. What is happening? What does the mitochondria itself need from the rest of the cell to make ATP? So go ahead and pause and go through this part. Hopefully you came up with this equation of carbon dioxide and water finally produces glucose and oxygen. This reminds you that the purpose of photosynthesis is to make food for the plant, not to make energy. That energy will eventually be used in the mitochondria, or in cellular respiration, I should say, to make ATP. But the ATP, excuse me, I'm sorry, the ATP made during the light reactions is all used to make glucose. And remember that oxygen is just a byproduct. It's not why the plant is doing photosynthesis. So here is your chloroplast. It's got that double membrane. Like it says, evidence for endosymbiosis. It has the thylakoid space, so the space inside these sacs. The thylakoids are these membrane sacs that are all stacked up. And then the stroma, really important for where the Calvin cycle is. This is just a summary to remind you that there's two sets of reactions, the light reactions and the dark reactions. ATP is made by the light reactions to power the Calvin cycle. Right? So that's the purpose of the light reactions. Again, you see a byproduct of oxygen, and then here's our most important end product. There are two, let's see what I'm gonna do. Yeah, so the light reactions consist of two electron transport chains. And I'm going to show you the Z scheme in just a second, but just let's focus here. So remember that we need water. Water provides the electrons for the electron transport chain. Light provides the energy to energize the electrons. And we have these two photosystems, photosystem 2 and photosystem 1. Okay. And so what's happening first is electrons are getting dropped off from water in photosystem 2. Change colors here. The electrons get excited and they go through this electron transport chain. And what they're doing is they're pumping hydrogen ions into the lumen of the thylakoid. Okay, stroma's out here. The electrons get dumped off in photosystem 1. They get excited from light again. Okay, and then they move and they produce NADPH. Those hydrogen ions from PS2 from PS2 go through ATP synthase and drive the formation of ATP. So in the stroma, you have NADPH and ATP being made. They're going to go and feed into the Calvin cycle. In the lumen is where you have the hydrogen ion concentration, or the proton motive force, or the electrochemical gradient, just like we talked about with cellular respiration. This is just to remind you what we were just drawing. This Z scheme reminds you that there's 2 electron transport chains in the light reactions. One is to make ATP, and one is to make NADPH, the electron carrier. Those are made in the stroma, and then they feed in. So here's ATP, here's NADPH. They feed the Calvin cycle. And so the key to the Calvin cycle is here's where your carbon dioxide comes in. And this is all happening in the stroma of the chloroplast. And we have this super important enzyme, rubisco. And rubisco takes carbon dioxide from the air and puts it into an organic molecule. So rubisco binds CO2, the carbon from the air, to an organic molecule. That's eventually going to allow us to make sugar, our end product. Okay. Rubisco allows us to take that inorganic carbon dioxide and make organic molecules. All right, so let's do, you've got 7 clickers here. So again, I would pause, go through the clickers, and then come back. So this question asks, a plant has a deficit in photosystem 1. It doesn't work. What would you need to provide the plant to keep photosynthesis going? So hopefully you remember that the end product of photosystem 1, that electron transport chain, is NADPH. ATP is made by photosystem 2. Water is the electron source. So that happens at photosystem 2 as well. And carbon dioxide doesn't come in till Calvin cycle. So you would need to give that cell some NADPH so that it could keep the Calvin cycle going. Here's a question. Why do leaves die in the winter and plants stop growing? So the correct answer is D. There's not enough light to power photosynthesis. So the plants can sense the decrease in light time. And so they stop making chlorophyll. They stop doing photosynthesis. They allow those leaves to drop off and kind of go into a dormant stage, right? Because they're not going to have enough power to make enough sugar to keep the plant growing through the winter. If you gave the plant an inhibitor of rubisco, what direct effect would that have on photosynthesis? So B is the correct answer. This is what rubisco is important for. It binds CO2 for the Calvin cycle. Which of the following is a reactant in cellular respiration and a product of photosynthesis? Hopefully you said oxygen. So remember, oxygen is required for cellular respiration and is produced when water is broken down in photosynthesis. This, excuse me, this reaction represents, sorry, my throat is just really clogged this morning. So this is aerobic respiration. We've got oxygen, that's aerobic, and we're going from glucose and oxygen, and we're making a lot of ATP. Which group is mispaired? So hopefully you recognized B was the answer. Water is not produced. Water is used in the light reactions. This is where water is produced when you add electrons to oxygen and oxidative phosphorylation. NAD pH is produced in the light reactions we just talked about from photosystem 1. This is true, and this is true. All right, one more for this unit. The Calvin cycle is not working well. What supplement could you give a plant to allow it to survive? So if the Calvin cycle is not working well, we're not making glucose. So you would need to provide glucose, the product of the Calvin cycle, to the plant so that it could go and use that glucose to make ATP through cellular respiration. Okay, that is the end of Unit 2. And I'm going to try to save this video for you.